Hello, welcome back to Dinosaurs. We're going to keep going through Module 1, these basic foundational concepts that we're going to build off for the rest of the course. We talked about fossil record last time. Today we're going to talk about taxonomy, how we sort of group organisms together into convenient bins for study. And then we're going to talk next week about evolutionary processes on various different scales. And so this is going to kind of introduce the topics today, and then we're going to discuss it in more detail next week. Before we do that, though, some announcements. All right, so let's review what happened, what we talked about last time. So I got a cat meow in the background here. So which type of fossilization is represented here? So this is wood, or rather it was wood. Now it's petrified wood. What is this process called? So take a look at those options there. It is permineralization. So over time, the wood fibers, the lignite, have decayed away, and they've been replaced bit by bit, slowly over time, by new minerals, in this case, probably agate. And it's actually replaced, reproduced the geometry so well that you can actually see the bark, you can see the splits in the wood, you can even see the tree rings. So this is not wood anymore. It's been fully mineralized, but you can still see some stuff. And this is what happens with dinosaur bones. The dinosaur bones generally are permineralized and lithified into rock and mineral, hard rock and mineral. It's not bone anymore, but we can still see the structures, usually. Okay, so what are the chances that a given bone enters the fossil record? So one problem is that bones are often broken up. This is an early hominin skull. You can see there's a lot of, the black is like an epoxy or a resin that sort of holds the broken pieces in place. This skull was found and they had to kind of cobble it together like a jigsaw puzzle, and some of the pieces are missing. It's very unlikely that anything gets fossilized at all. We're very fortunate that we have any fossil record to look at. What are the chances that a given bone enters into the fossil record? And again, this is like a general rule of thumb. And the answer is 1 in a billion. So again, thinking back to last lecture, if you take all of the humans in the United States today and attempt to fossilize them, we don't want to do that, but we would end up with about 1/4 of one skeleton. Not even enough bones to assemble an entire human skeleton, only about 1/4. So that's how unlikely it is that things are fossilized. It's pretty amazing to think that we have any fossil record at all. And so just bear in mind that there's a lot of the gaps. The fossil record is actually more of a lack of record, and paleontology is trying to reconstruct everything from that very incomplete story. Okay, so let's get into the lecture for today. So we're talking about taxonomy for today. So what is taxonomy? It might not be a word you've heard before. You've probably heard it in biology, but so taxonomy is sort of the laws of order. So we're trying to take all this random chaos of nature. So this is an illustration of Tree of Life. You see all these different critters on here. We're trying to take all these different critters and we're trying to, and plants, and we're trying to put them into buckets and bins based on their similarities. So why might we want to do this? Well, it's very helpful as scientists if we have like a common language, that if I say the word mammal, you know that it refers to this certain group of organisms with this certain characteristic. If I say the word reptile, that has other implications. If I say dinosaur, that means a very specific type of reptile with very specific characteristics that you should know from the earlier lectures. And if you don't, remember, you can always go back. These are videos, you can play them at your leisure, watch them multiple times because I'm so charismatic and inspiring on the videos. Pause, rewind, watch 100 times, share with your friends, share with your family, yada, yada, yada. But anyway, remember though, that all the taxonomy that we're going to talk about today, all of the different ways of classifying organisms, all of these groups that we assign organisms to are inherently artificial. They're man-made. In nature, there really is no such thing as a mammal. It's a word and a bin that we created to put these things into and store them. the only thing that actually really truly exists is species. In nature, a species are individual organisms that can interbreed with each other and have viable offspring. All of the higher level genus, family, all the way up to phyla, those are artificial groups that we've made. Now, just because they're artificial and man-made doesn't mean they're not useful, but it does mean that they can be flexible. And we have made the choice to group things in this way. And there are a bunch of different ways of doing that. And there, in some cases, there's some controversy around maybe how we should do it and where we should draw the lines and what goes in what bin. But so bear that in mind. There's this famous phrase here, God created and Linnaeus organized. So Carolus Linnaeus was the one that first started trying to do this, is the kind of father of taxonomy, the one that started trying to group these things up. And we still kind of, to this day, utilize the system that he developed. So all of the biodiversity of the planet, so bio means life, diversity means different types, all the different types of life on this planet are assigned into groups. These groups are called taxa. And systematics is the science of the diversity of all of these organisms. And so there's this hierarchy here. We start at the top at kingdom. So kingdom, and so here's a leopard. We're gonna kind of walk through the hierarchy of how we classify a leopard. So a leopard is in the kingdom Animalia. It's an animal. Some of the other kingdoms, there's the plantae, there's the fungi, there's the archaebacteria, there's other kingdoms. Leopard is an animal. It goes in Animalia. That's a very large bin. All of the animals that have ever existed fall into Animalia. All reptiles, all amphibians, all mammals, all humans, all primates, all sea invertebrates, they're animals. All of the variety of animal life goes into Animalia. This is a very large bucket. Getting a little bit more specific, the phyla for this is chordata. Chordata are things that have a backbone. They're the vertebrates. We are also in chordata. And so us and this leopard are in chordata. The next bin down are not only things that are animals that have a backbone, but also mammals having a general body covering of fur, warm-blooded, mammary glands. Again, we are also mammals. The order, again, getting a little bit more specific, is carnivora. So things with sharp, pointy canine teeth for ripping and chewing and devouring meat. The leopard is in carnivora. Getting more specific than that, the family is Felidae, the felines. The genus is Panthera. And the species is Panthera pardus. Only once you get all the way down to the species level do we know exactly what organism you're talking about. And again, Panthera pardus, these leopards, they can only successfully, viably interbreed with other leopards. And so that's the only one of these bins that's real, that has natural significance. All these other bins are things that we've sort of artificially made. Some of them are more useful than others. So like, for example, chordata, there's the invertebrates. That's a kind of a waste bucket taxa that says things that it's not. We'll talk about that in just a second. But you do need to remember this hierarchy here. And so one way to kind of remember it is this mnemonic here. I always like to make mnemonics. It helps remembering complicated things pretty easy. But so at the very top, there's domains. We're not going to worry about that. But so kingdom, phylum, class, order, family, genus, species. The way that I remember it, the way that my biology teacher taught me to remember it, King Philip came over for good spaghetti or greasy spaghetti. I've never had greasy spaghetti. I don't think I want to have greasy spaghetti. I do want good spaghetti though, so that's what I use. But if you can remember greasy spaghetti better, go for it. And then the important thing here is that when you take the genus and the species together that makes the binomen. Bi means two, nomen means name, two names. So like, for example, Stegosaurus ungulatus, that genus Stegosaurus incorporates all of the stegosaurs, and ungulatus is a particular type of stegosaur. They're the roofed, hoofed stegosaur. We'll talk about that stuff when we get to the stegosaurs, but for example, us, Homo sapiens, that's our genus Homo, and our species Sapiens. There's also Homo erectus, Homo ergaster, earlier hominids that fall into that genus, but only us is that species. So keep that in mind. We do need to remember this. King Philip came over for good spaghetti, kingdom, phylum, class, order, family, genus, species, getting more and more specific as you work your way down. Okay, so we've got these bins, or we're trying to create these bins anyways, and we're trying to lump organisms into them. How do we do that? How do we start with all of these various critters? How do we start making buckets for them? Well, a lot of early classification schemes were based on like sort of like practical similarities, or just like very high level superficial similarities, like how they're built, what they look like, how they function. So for example, poisonous versus non-poisonous plants was a bucket that was used. These are the poisonous plants, these are the non-poisonous plants. All poisonous plants go in this bucket, all non-poisonous plants go in this bucket. Now, with a modern view, we know that, well, there's a lot of different ways, a lot of different branches on the tree of life that creates plants that are poisonous. Not all of them are directly related to each other. Many of them are very distantly related or only tangentially related to each other. And so this is maybe not the best way to do it, but it is something, you know, it works, it's a bin, but maybe there's a better way of doing it. Another thing you can do is like kind of like functional morphology. What does the animal do? How does it behave? So like if you're gonna do that, flying fish kind of jump out of the sea, and they actually have, their fins are modified to be essentially rudimentary wings. They're actually able to kind of glide for short periods of time. In fact, people are often kind of hit with them flying through the air. They fly, they glide just like birds do. In fact, here is one being chased by a bird. They're sharing the sky together, in this case, not so happily. Are flying fish birds, or should they be in with the fish? Probably the latter. They're more closely related to fish than they are with birds. But superficially, they do this kind of same thing. They fly through the air for brief periods of time. That's a group that's probably not the greatest, but it's something you could do. Or think about like armadillos. Armadillos are those little plated dudes that can roll up into balls. Turtles have, again, the scutes, the plates to build their shell. Armadillos and turtles kind of behaviorally do sort of similar things. They're these little moving little tanks, but they're very different from a lot of other perspectives. And so maybe grouping them together is not the best way either. So obviously, because again, there's no right answer here, only species actually really exists in nature. So you can kind of go back and forth about, is this actually the right way to do this? How should we classify? What's the proper way to classify? So the first way that we tried this was with what's called evolutionary taxonomy. So Darwin, he wasn't the first to do this, but Darwin noted that Linnaeus's classification, so the Linnaean classification system that King Philip came over for greasy spaghetti, good spaghetti, it closely mimicked evolution. So when you started grouping these things into those phyla, when you started grouping things into families, when you started grouping things into genuses, it closely mimicked evolution. And so it was basically creating these natural groups that sort of reflected a shared evolutionary history. Unfortunately, in a lot of ways, these groups that we've put in become sort of this awkward mix of morphology, like shape, how they're constructed, ecology, how they behave, which are strongly linked with each other, and phylogeny, their evolutionary history, their ancestry. So like the one big problem that we have here is a lot of the groups that are defined in evolutionary taxonomy, a lot of groups are defined by what they're not more than what they actually are. So like chordates are defined by having a backbone, but the invertebrates are defined as not having a backbone. So when you start defining something by what it's not, you're lumping things that are very different from each other, or at least you can. So that's problematic. We also created some wastebasket taxa, where there's like this really crazy organism. We don't necessarily know where it goes, but there's others that are kind of like it. We're just going to make this group of like, these are things that we don't really kind of know what they are. So like platypi, like the monotremes, the egg-laying mammals, we don't really know what these things are. They're very different from other mammals. Let's make this group that's just them. Now, they may or may not share a common evolutionary history, and that can be problematic, defining things that way. So this is kind of the way that we usually do it. This is the kind of like high school level biology classification scheme. Another approach that was kind of tried as computer power was increasing, and as we were becoming more and more reliant on technology, there was this whole new field called numerical taxonomy, sometimes called phonetics or taximetrics. People were noting that a lot of these groups that the evolutionary taxonomists are making were kind of arbitrary, and it required this art, and certain people would classify certain ways, others would classify another way. It was very subjective, it was very opinion-based. There was a lot of argument back and forth about, no, I'm right about what bin it goes into. No, I'm right about what opinion. So this was sort of an attempt to there must be a right answer. We can figure this out. We've got numbers, we've got computers, there's an answer, let's do it. So numerical taxonomy is based entirely on statistical similarity, lumping things that are statistically similar to each other, and separating out things that are statistically different from them and kind of making these groups. So to do that, you have to figure out, well, okay, we're gonna see how similar things are based on what? What are we gonna measure? What are we going to assign numbers to crank through this thing? And so they came up with this concept of operational taxonomic units, or OTUs. So you come up with something and you measure it, like number of limbs, or wings or not, something like that. It's like a binary or a number, something that can be quantified. You plug all of those into a computer. The computer cranks and it spits out an answer. Now, the computer is completely unbiased and objective. It's not opinion-based. The computer doesn't have opinions. It does what you tell it to do. Is that actually an unbiased result, though? Well, humans, objectively, opinion-based, had to decide what those OTUs were. What were we going to measure? What is important? What do we value? And so we've plugged our opinions into the algorithm, and so it doesn't really help. It's still an art. It's still not objective. It's still strongly opinion-based. It just has sort of this illusion that it's quantitative and objective and unbiased, but garbage in, garbage out. If your entire algorithm is based on your opinions, the end result is going to be a result of your opinions. And so it doesn't really help that much. And so it's not really used all that much today, but it's a very simple, quick, easy thing to do. Obviously, there was a lot of struggle when this came out, and numerical taxonomists come on the scene and they're like, we plug it in the computer, we get the right answer. All you evolutionary taxonomists are wrong. So there was actually a back and forth at a meeting, and one of the traditional taxonomists said, you mean to tell me that taxonomists can be replaced by computers? Like, you can just plug your stuff in the computer, run the numbers, and get the right answer here, and replaces everything that I do for a living? And then the numerical taxonomist said, no, some of you can be replaced by an abacus, so the little thing with the beads. So this is the level of animosity that there was between traditional taxonomists and numerical taxonomists. Like, computers were the wave of the future, like, get out of the way, traditionalists were coming through, we've got the right answer. And it turns out that it was sort of a passing fad. It didn't solve a lot of problems. So a more modern approach, sort of the extreme from phonetics, is so phonetics kind of lumps morphological characteristics, shapes, sizes, features. Cladistics bases only on the evolutionary history, and traditional taxonomy kind of does a little bit of both. We're going to be using a lot of cladistics in this class. I'm just going to define it a little bit quickly here. We're going to spend a whole lecture on it next week, but cladistics is classification that's based just on evolutionary relationships, also called phylogeny. So there's a lecture next class called cladistics/phylogeny. Phylogeny is the evolutionary history of an organism. What steps along the way were there to make the organism that we see today. It ignores morphology, and it ignores ecology, behavior, similarity, unless those similarities result from being from a common ancestor. So if two organisms happen to fly, if that happening to fly results as that their common ancestor flew, then they're in the same clade. If they happen to fly because they just happen to evolve it independently, and we'll talk about that in just a second, then that's not a clade because it arrived at it differently. So the fact that they both fly doesn't matter if their common evolutionary ancestor didn't fly. So this is a cladogram, a relatively simple cladogram. And so the way that you read this is we start at the bottom, the most primitive. That's a word that has a little bit of baggage to it. I don't like using that, but it's the easiest way to express the concept. Up to maybe more advanced, and again, advanced has baggage too. Just remember that all animals are equally viable for their niche. They have evolved to fill their niche perfectly. A salamander is not less advanced than a leopard just because it's a little bit farther down the cladistics chain. A salamander does what a salamander does, and it does it very well. A leopard does what a leopard does, and it does it very well. All of these organisms are perfectly fine on their own. The fact that one is farther up the chain, the progress, that's kind of a misnomer, and we want to kind of avoid that. But anyways, I just said it, so let's start down here. So the lancelet, very basic, very early, it has a notochord, doesn't really have a true spinal cord. And then along here, we develop a vertebral column, a spinal column. And a lamprey has a spinal column. So that's the first organism that branches off here. Lamprey do not have hinged jaws. The next step on the evolutionary chain is the development of hinged jaws, and tuna have hinged jaws. The next step up on this particular cladogram is 4 walking legs. Tuna do not have 4 walking legs. Salamanders do. And so the way you kind of read this cladogram is at this point on the line here, 4 walking legs develops. Everything before this part doesn't have 4 walking legs, and everything after does. So let's go to the next step. Salamanders have 4 walking legs, but they do not have amniotic eggs. They have to lay their eggs in water. They're amphibians. Turtles do have amniotic eggs. They lay eggs on land, or at least they can. They're these capsulated eggs. Turtles have amniotic eggs. They do not have hair. Leopards do. One thing you might notice is that it says, here's amniotic egg. Everything before should not have an amniotic egg. Everything after should. Do leopards lay eggs? No, they don't. But humans and other mammals, the embryo develops from an egg inside the organism. So in some cases, these similarities are a little less obvious, but they are there. We do use eggs. It's just not in the traditional sense of a laid egg, it's internal. So this is a cladogram. We'll walk through this in depth, how to read these, but in general, it kind of goes up, and those that share similarities are behind, and new things are up here. So again, like, what characteristics do we use? We use characteristics that have evolutionary significance. So there's this concept of derived characteristics. So like, for example, in mammals, mammals having hair, mammals having mammary glands, mammals being warm-blooded, those are specializations that define that group. Those are derived characteristics, characteristics that were formed in the mammals and don't exist in the previous groups. Primitive characteristics are characteristics that are present in the previous groups. So like if we go back to that cladogram on the previous page, all of those things had a spinal column. A spinal column is not a unique characteristic of mammals, but again, hair, warm-bloodedness, that is. So again, each node here, each branch represents a common ancestor that has that trait. So like, again, starting out with a lamprey, lamprey is going to be picked on quite a lot. They're a relatively early developed organism, a lot of primitive characteristics. The next step along here is development of jaws and vertebrae. Sharks have those, lamprey do not. Moving along, forelimbs and lungs, frogs have that, sharks do not, lamprey do not. Everything up this way does, or at least did at one point. Then we get hair and mammary glands, the mammals, cows have that, monkeys have that, humans have that. We get up here opposable thumb stereo vision, monkeys and humans have that, cows don't, frogs don't, sharks don't, lamprey don't. And then we get to no tail. Humans do not have a tail. The tail is diminished, only our little tiny tailbone. Monkeys do. And so that's how you read a cladogram. And again, side of opinion-based, what characteristics are actually useful here? What creates the cladogram? Again, that's where kind of the art comes in here. And the simplest answer is the best answer. And we'll talk about all that next week. There's a couple of things that we need to know. Again, when we're trying to do cladistics, we're trying to group things that are based on shared traits that are the result of a shared evolutionary history, a common ancestor. And so these things are called homologous structures. So homologous structures, they're very similar in structure, they're very similar in location, but they're not necessarily the same function. And this shows that there was an evolutionary relationship. So for example, the 5-digit human hand, the 5-digit dolphin flipper, the 5-digit elephant foot, all share 5 digits. Birds are a very highly modified 5-digit forelimb, but they all share this 5-digit past. Why? Because our shared evolutionary history all comes from the first organism that crawled up on the land, the earliest tetrapod, 5 fingers. Now, if we look at analogous structures, they're very similar in function. So they're used for the same purposes, but they're not in structure or location, and there is no evolutionary relationship. So for example, birds' wings and the flies' wings, they serve the same purpose, to propel them through the air, but the internal structure of a bird's wing and a fly's wing are entirely different because their evolutionary history is entirely different. So if we start lumping things based on analogous structures, that's probably not the right thing to do. Their similarities are superficial at best. If we look at homologous structures, even in the cases where they might not look exactly the same, it shows that they evolved from a similar ancestor. So let's take a little bit more detail. So here's homologous structures. Again, similarity in structure, location, due to shared ancestry. Here is a human hand, 5 digits, 2 arm bones, the ulna and the radius, the humerus, that's the structure of the human arm. Look at a cat paw. Cat paws, of course, don't look exactly like human hands, but cat paws have 5 digits. They have the added toe beans and toe razors, but structurally it's very similar to our hand in the number of bones they're positioning, with a little bit of difference. Again, 2 arm bones, 1 humerus bone, roughly the same structure. A whale fin is radically different from a human hand, but they have 5 digits, the 5th one very much reduced. The 2 arm bones, the ulna and the radius, are substantially reduced, but it's there. And the humerus is substantially reduced, but it's there. The same number of bones, basically. The same basic morphology, but modified to serve a different purpose. And then bats are another example. Their fingers are highly elongated to support the membrane of their wings, but they still have the 5 digits, the 2 lower arm bones, the 1 humerus. Why? Because all of these are land-based tetrapods, all land-based terrestrial organisms. Whales eventually went back to the ocean. Bats eventually went up to the sky, but they all started on land, their shared ancestors, from when Tiktaalik in the Permian decided to crawl out of the ocean. It evolved from a fish that had a fin. Tiktaalik itself had fin with wrist to kind of move around a little bit better on the land. And then the very earliest amphibians that were actually developed for on land had a different modification where they actually had a limb for moving around. All land-based critters, including the dinosaurs, came from this common ancestor. And so dinosaurs share that as well. All land-based tetrapods have these features because they originated from a common ancestor. Now, analogous structures are different. So analogous structures, the similarity in the structure and location of a feature is due to solving a common problem. So evolution over time is trying to solve a problem. Like, for example, in this case, evolution is trying to solve the problem of I'm a water-based creature. I need to glide through the water using as little energy as possible so I can conserve my metabolism. I don't have to eat as much. What you'll see is that these organisms converged on a very similar shape. Superficially, they look real similar. They've got that bullet-shaped streamlining. They have fins to propel themselves. They have teeth for preying. But an ichthyosaur is a reptile. It's not a dinosaur, it's a reptile. Remember, dinosaurs are a special type of reptile. A shark is a fish, and a dolphin is a mammal. These are very different organisms. They have no evolutionary relationship, at least it goes way far back. Their common ancestor is way far back. They're very distantly related evolutionarily. They've converged on the solution here because it's the best solution to the problem, getting around in water. If you look in detail at the things that sort of look the same, they're really not. The fin of an ichthyosaur, shark, and dolphin are very different from each other, even though they sort of do the same thing. They do the same thing, but the structure is different. Dolphins are related to mammals. Dolphins are mammals. They have the 5-digit flipper. Fish, sharks do not. Ichthyosaurs, they do have a distant related to the 5-finger because they came from land-based, but very different flipper from a mammal. Now looking at flying things, again, nature has to solve this problem of I'm a creature that wants to fly through the air. How do I maintain myself in the air? Well, I need some kind of wing to maintain loft or just a basic gliding. Again, if we look at pterosaurs, not a dinosaur. They're a very specific type of reptile. Dinosaurs are a very specific type of reptile. They're sort of close-ish related. We'll talk about how close a little bit later, but they're not dinosaurs. Remember, the things that define the dinosaur are different. Go back to lecture 1B if you're confused. A mammal wing does the same thing. It keeps it aloft. A bird wing does the same thing. These are all wings, and they look at a first glance to be very similar. Okay, a wing is a wing is a wing. But looking at the mammal wing, the bat wing, 5 fingers, the bat is using all 5 of its fingers to support the wing membrane. And it's even actually connected to the leg as well. Pterosaurs are using only 1 finger, 1 greatly elongated finger, to support their wing membranes. There is this big argument about whether pterosaur wings actually attached to the legs like bats do. It appears that they did not. So why would it be such a controversy? Well, again, usually only the hard parts are preserved. The soft wing membrane is incredibly delicate, very unlikely to be fossilized. But we do have some evidence now that they probably were not connected to the legs like bats. And then a bird wing, the fingers and the wing don't really do anything to support the wing. It's the feathers that do all the work. And so if you've ever eaten chicken wings, like chicken wings do not have these big, long, elongated bones. They've got really stubby bones. It's the feathers that come off of the wings that do most of the work. So even though they look, okay, a wing is a wing, they're very, very different. Why? Because they don't have an evolutionary relationship. Reptiles, mammals, and birds all evolved on separate evolutionary paths. And so their morphology, their structures are different. They're analogous because they perform the same function, but structurally they're different. If you're still struggling with that concept, this is a really good video on the topic. I'm not going to play it here. I'm not going to go full Inception video inside a video, but it's here. So I do encourage you to watch it. All right, so again, convergent evolution. When we're talking about grouping animals together based on similarities, which is what we're trying to do to make these bins, when we're trying to figure out what's evolutionary relationships and what is just happenstance morphological similarities, just shapes that sort of look like each other, it can be very tricky. So again, these examples here, a fish, a dolphin, a mammal, superficially they look very similar. They are not related to each other evolutionarily. It's just that they've all evolved independently towards a common efficient and very successful form. If I want to swim through the water, this streamlined bullet shape is highly efficient. If I want to swim through the water, the fins is a good way to do it. One thing you'll notice is that fish fins are upright. Ichthyosaur fins, the tail fin happens to be upright as well. Dolphins is off to the side. Solving a similar problem slightly differently, the morphologies are different, but it can trick us. If we're just looking at similarities, you might lump all these things together in like the swimmers group. They're not related to each other evolutionarily. It's convergent evolution, evolution converging towards the same solution multiple different times. Saber-toothedness is another good example of convergent evolution. There are saber-toothed kynodonts back in the Permian. There are saber-toothed tigers, saber-toothed cats in the Pleistocene. Developing saber teeth is a good way to kill stuff. And so it's evolved independently several times throughout geologic history. Again, this is a good video that walks through divergent evolution, things kind of radiating outwards towards different forms, versus convergent evolution, sort of things converging on the same solution to the same problem from different evolutionary lineages. Again, not going to go Inception, video and video, watch it on your own time. Okay, so let's compare classification methods. So there's the traditional classification scheme, again, that King Philip came over for good spaghetti, kingdom, phyla. That's relatively clean, relatively uncomplicated, but in many cases, it's not quite as useful because sometimes we have to lump things together that maybe shouldn't be. So for example, humans, the traditional classification puts a superfamily of Hominoidea, and the superfamily Hominoidea consists of all of the apes, and another family that's all of the humans. One problem with this is that this family Ponchidae, apes, the only structural difference between humans and apes is that the apes aren't humans. And so the only real difference between Hominidae and Ponchidae is that humans aren't in this one. And so Ponchidae is this awkward kind of wastebasket where, okay, yeah, they're all hominoids, but these ones are the humans and these ones aren't. Why do we draw the line there? Why are humans so special? What's special about humans that we need our own separate family? Well, our own vanity for one, but this is the traditional classification, and there's some problems with it in that it creates these kind of awkward divisions, these awkward wastebaskets, again, like vertebrates versus invertebrates. Invertebrates is a wastebasket for things that don't have backbones. It's sort of an artificial character based on what they're not rather than what they are. And so this is a cladistic classification where, again, we have superfamily Hominoidea. We have a family of the gibbons. There's a tribe for the orangutans. There's a tribe for gorillas. There's a tribe hominini which we share with both chimps and us. There's a subtribe, the Panini, I don't know how to say it, is chimps, just the chimps. Then there's the genus Pan, Homine, and genus Homo, which is us. And then Homo sapiens is humans. Homo neanderthalensis, Homo erectus, Homo ergaster, lots of other Homo species in that genus. So this is a little bit more useful. These bins are sort of real. We see the difference between gibbons, orangutans, gorillas, chimps, and humans, and they all have their own special bins. We're not lumping all the non-human apes together, even though they're somewhat different from each other. And we're not just making humans like its own special thing, at least not at the same level as all the other apes. But you can see that it starts getting pretty complicated. It's a lot longer. In general, for this class, we're going to use a lot of cladistics. We're going to talk about dinosaur clads. We're mostly going to be using this. You'll see a lot of the genus species binomins, like Tyrannosaurus rex, Tyrannosaurus being the genus, Rex being the species. There are a lot of Tyrannosaurids that fall into the genus, only one Tyrannosaurus rex. We're mostly going to be using these clads. So this can be a little frustrating. It can be a little confusing. So which of these methods are we going to use? Again, in this class, we're mostly going to be using cladistics, but if you can use the evolutionary taxonomy traditional approach, the Linnaean approach, or you can use the numerical approach, or you can use the cladistic phylogenetic approach, which one are you supposed to use? And one answer that's probably going to get frustrating to hear throughout this class and through any science class is it depends. There is no right answer. None of these classification schemes is any better than the others. It really depends on what you're more interested in. Traditional taxonomy kind of lumps things based on their superficial similarities, their behavioral similarities, and in some cases that happens to just coincide with evolutionary similarities. Cladistics and phylogeny sort of look at just the evolutionary history. And so it depends on what are you more interested in. Are you more interested in behavior, or are you more interested in the evolutionary history? It's incredibly hard to lump dinosaurs together based on their behavior because we don't really know. Again, there's the fossilized hard parts. Soft parts usually are not preserved. Behavior is only really occasionally preserved in trace fossils. It would be very hard to use traditional taxonomy on dinosaurs. And so what we do is we generally use the evolutionary relationships. And so that's what we're mostly going to be using is the clades of dinosaurs that are similar based on their shared ancestry. And so that's mostly what we're going to be doing. So it'll get a little frustrating because it doesn't quite have that same structure, but it's a little bit more useful in terms of dinosaurs. So cladistics can be a lot more organized. Again, if you think back to the way the lectures are structured in modules 3 and 4, like we're going to talk about the sauropoda in one lecture. We're going to talk about the Pachycephalosauria. We're going to talk about the theropods. Those clads each get a day because they're like their own group. That's like a consistent group, a clad. It's a little bit more organized, a little more convenient, shared ancestry. That's mostly how we're going to talk about this. So that's all I've got for today. I hope you enjoyed that. Make sure you're posting up on the discussion board. Make sure you're following along. Make sure you're doing the assignments. Make sure you're completing a checklist. It's Friday for me. I don't know if you're watching this on Friday or not. If you are, have a good weekend and goodbye.